Good morning, everyone. It is really my great honor to be able to moderate this morning's panel discussion. We have a big group. We have 7 panelists. So on behalf of all the panelists, I would just like to thank you all for being here and to thank especially our organizers for this opportunity to speak about this important topic. 谢谢你们主办单位给我们这个机会来谈谈这个重要的主题。So, So this morning we'd like to shift perspective a bit and focus our attention on what the stem and stripe rust pathogens are doing. There we go. When they are not living on cereal hosts. To help set the mood for this, I'd like to show you a short film that we recently produced as an educational piece by the BGRI. Destroying harvests for millennia, the stem rust pathogen of wheat is a sophisticated organism that continues to this day to threaten the security of global wheat production. The emergence of the UG99 group of virulent stem rust races underscores the need to better understand the mechanisms of resistance in wheat and to develop new varieties resistant to current races of stem rust. But the emergence and rapid spread of new forms of virulence also prompts more fundamental questions. How do new virulent races arise in the first place? And what factors contribute to the rapid and ongoing evolution of this historic pathogen? Stem rust of wheat is named for the powdery, reddish-brown, rusty-colored blisters or pustules that develop on the stems and leaves of susceptible wheat lines grown under conditions conducive to infection. This telltale reddish-brown powder found on infected wheat plants are spores of the fungus Coccinia graminis, the organism that causes stem rust. A single pustule can produce tens of thousands of these spores, called Aeciospores, with each one capable of infecting a wheat plant and producing yet another pustule. Carried by the wind, perhaps to a neighboring plant, perhaps to another wheat field hundreds or even thousands of miles away, a microscopic Aeciospore can land, germinate, and cause new infection. Once inside the host tissue, the pathogen extracts nutrients from the wheat plant's cells, grows, and eventually produces a new generation of Aeciospores in such abundance that the plant's tissue erupts, releasing the new spores into the air to repeat the infection process elsewhere. On and on the cycle goes. With roughly 2 weeks from infection to sporulation, this cycle of Aeciospore multiplication can rapidly produce epidemic levels of disease. A severe stem rust infection in the field is a dramatic and devastating sight, but it is not the whole story. An important point about the Aeciospore cycle of infection is that it is a clonal method of spore production. Random mutations, genetic copy errors, and potentially rare cases of asexual hybridization aside, Aeciospores infect wheat tissue and produce exact copies of themselves. For a highly virulent race of stem rust like UG99, it is this cyclical process of spore multiplication that causes epidemic levels of disease within a season and necessitates the development and deployment of resistant wheat varieties. What a perfectly clonal cycle does not explain fully is the emergence of new virulent races. For this, it is instructive to shift our attention away from the wheat host plant and to continue to follow the pathogen. As the growing season comes to an end and wheat plants begin to set seed, senesce, and die, the stem rust pathogen does something remarkable. Instead of producing more Aeciospores, it begins to produce black overwintering spores called teleospores. Although teleospores cannot infect wheat, they are important for 2 reasons. First, they can survive in a dormant state on dead wheat stubble, something the more sensitive Aeciospores cannot do. And second, It is in the teleospore stage that the two haploid nuclei fuse together, the first step toward creating new genetic variation and possibly new virulence. A period of cold temperatures is part of the environmental requirement to break the dormancy of a teleospore, at which point it can germinate and produce through meiosis haploid spores called Basidiospores. Incredibly, these Basidiospores infect not wheat, but certain species of the completely unrelated woody perennial plant genus Berberis, or Barberry. Transported by the wind to barberry plants near wheat fields, Basidiospores can germinate, infect, and create blisters called spermagonia on the upper surface of barberry leaves. These blisters produce reproductive cells called spermatia, able to fertilize receptive cells called hyphae inside compatible neighboring spermagonia. In this step of fertilization, entirely novel genetic combinations are achieved. Once fertilized, the hyphae grow down through the barberry leaf, where they eventually produce stunning reddish pustules on the underside of the leaf, marked by numerous peach or orange-colored tube-like structures called aecia. When exposed to moisture, these aecia, quite visible to the naked eye, forcefully eject the next spore form of stem rust, Aeciospores. Carrying entirely new combinations of genetic material due to the cycle of sexual recombination on the alternate host, Barberry, these microscopic Aeciospores can once again directly infect wheat, producing the brownish-red uridinospores that are the hallmark of this disease. The complex and amazing life cycle of Puccinia graminis suggests that achieving durable resistance to wheat stem rust depends not only on the critical task of developing more resistance in the primary host plant, wheat, but also on investigating the prevalence and functionality of the alternate host, Barberry, and utilizing knowledge of the complete life cycle of the stem rust pathogen to limit the emergence and survival of new virulent races.
All right, so now we're all in the mood for alternate host discussions. Okay, so continuing. All right. So just as our interest in breeding for resistance to the rust is not new, our interest in the alternate host is not new either. But while efforts to discover and deploy new resistance genes have made great strides since the discovery of UG99, rekindled interest in the alternate host has lagged behind. From a crisis management perspective, with the UG99 race group, this makes sense as understandable. But in terms of looking ahead and working toward an increased durability of resistance to the wheat rust, history suggests that in some regions at least, we may benefit from trying to control the emergence of new virulence races in the first place, in addition to breeding for enhanced resistance. Though the connection was not formally made until 1865 with the work of Barberry, the negative impact of barberries growing in proximity to wheat fields had been recognized for hundreds of years, as evidenced by barberry eradication laws in France in the 1600s, and the 1700s and 1800s saw similar laws in the New World and across different countries in Europe. In the United States, following devastating stem rust epidemics in 1904 and 1916, a nearly 60-year barberry eradication program was carried out. During this program, an enormous amount of resources was invested, not only in the direct eradication of the plants themselves, but also in educating the public more generally about the threat posed by barberries in wheat-producing regions. And these are some of the materials from that time. In a paper summarizing the US eradication work in the 20th century, in which over 100 million barberry bushes were destroyed throughout the north central plains. Dr. Alan Rolfs concluded that barberry eradication led to 4 measurable results. The first 2, delayed onset of disease and reduced virulent inoculum in the spring. These first 2 relate to the fact that barberry can harbor local early sources of inoculum. The second 2 relate to the fact that barberry provides a mechanism for increasing the diversity of the pathogen population. By both decreasing the quantity of early season inoculum and by stabilizing the pathogen's races, barberry eradication led in the United States to measurable increases in the durability of diploid resistance genes and was an important contributing factor to the decades of stem rust control now threatened by the emergence of the UG99 race group. So with a historical lesson like that in mind, and knowing now that barberry can also serve as an alternate host for wheat stripe rust, it's interesting to step back and take a more global perspective. Many of the known hotspots for novel race emergence for both stem and stripe rust, places such as East Africa, Central and Western Asia, Southwestern China, the Pacific Northwest of the United States, these are also areas for which there really is no history of systematic surveillance or control of the alternate host. Given the highly diverse group of endemic barberry species found on every continent except Australia, and given the rapid emergence of novel races of these historic pathogens from certain regions, several basic research questions present themselves. First of all, where's my first research question? Come on, there we go. First of all, what is the distribution of alternate hosts relative to wheat production around the world? And second, what role do these populations play, if any, in the emergence of new virulent races? So we are very fortunate today to have with us a panel of 7 scientists from around the world whose research considers these questions. While there were a few talks in the past few days which touched on the alternate host and various questions about the alternate host, this morning now we really have an opportunity to focus on this topic and to focus on this essential question. Do alternate hosts undermine our efforts to achieve durable resistance? As a general structure to the presentations this morning, we're going to begin with Drs. Waniera, Gaetane, and Skolotneva discussing their survey work on barberry as it relates to wheat stem rust in their regions. Then Drs. Berlin and Kong will widen the discussion to other Puccinia species, including wheat stripe rust. Dr. Nazari will then discuss the potential role of barberry in Central and Western Asia, the center of diversity of the genus. And finally, Dr. Yu Jin will wrap up with a discussion of our current gaps in knowledge. So after all the panelists present, we'll hopefully have 15 or 20 minutes remaining for questions and discussions. And with that, Ruth, come on up. And with that, I'd like to give the floor to Dr. Waniera from the Kenya Agricultural Research Institute in Njoro.
Thank you, Iago. I'm going to share with you the Barberry story in Kenya, what we've experienced and what we've gone through and how far we are. Yesterday, Dave gave us a presentation, and from that slide, all along, we've been having 7 variants of UG99, but as of yesterday, we learned there are now 8 more. And East Africa being a hotspot, we are wondering whether UG90, I mean the alternate host could be found in Kenya or in the East African region. So the present and rapid emergence of UG99 and its derivatives, starting with the SR31, followed by SR24, and then SR36 and others, suggests that sexual recombination may play a role in the evolution of PGT in East Africa. So while we are thinking about the sexual recombination, we went ahead, or we are asking ourselves 3 questions, or rather about 3 questions. The first one is, are there Berberis in East Africa? Then the second one is, if they are they susceptible to the serial rusts? And the third one is, do they function as alternate hosts under natural conditions? And from the film, we've just seen we've seen the life cycle of wheat stem rust. And now, given the biology of the wheat stem rust, we wanted to go ahead and find out, do we have the Berberis in Kenya or in East Africa? So we went to the National Museums of Kenya, and indeed, the records showed that there are Barberry species in East Africa. And so what we did, we started hunting for the Barberry, and we are hunting for bushes that we didn't know how they look like. And we sought, so far we've been to 6-7 sites. And out of the 7 sites, and out of the 7 sites, 2 sites, Mount Narok, that's the second one on the slide, and Mount Kenya, we found Barberry bushes. But the Barberry bushes from Narok, we couldn't identify the Aecia in the lab. We only identified Aecia on samples from the Mount Kenya region. And while we were trying to look for the Berberis, we also found out that they are medicinal. The fruits are also eaten by children. And from this slide, from that picture, from this picture, these are Barberry bushes that have been dug for use. I mean, the communities dig them up and they use the roots as medicine. So we also sought the assistance of the local communities by using local names to show us where the Barberry bushes are. And we also used the experts from outside the wheat rust community. So the leaves that we collected that had the infection, we carried them back to the lab, that is in the greenhouse back in Njoro, and we tried to carry out inoculations to find out whether the Aeciospores can infect the wheat plants or the cereal plants. But this was not successful, and we thought it could have been contamination, Njoro being one of the wheat growing areas, or rather the environment was not conducive for sporulation infection to take place. So we thought of an alternative, and we went to another carry center, which is a site where wheat is not grown, and there are no Barberries around that area. So we also tried to carry out the inoculations, and still we couldn't succeed. And again, the question we asked ourselves, was it because of the environment, or what was it? So we haven't gotten the answer to that. So at the same time, as much as we try to do the inoculations in Njoro and away from Njoro, we also try to send the Aeciospores, the infected leaves with the Aeciospores, to the Serial Disease Laboratory in the US, but they were I mean, it was not successful in a way that by the time the leaves were arriving at the lab, the Aeciospores were not viable. So we sort of got stuck. And then from our experiences, we think maybe the environment is not right. We are moving the plants from the environment, or rather the infrastructure we are using is not up to date. So as a way forward, we are trying to suggest that if we got resources, we would do submolecular work for diagnostics locally, and that would ease to work quickly on the samples that we have collected from the field. And again, at the same time, we also would like to capacity build, in-country capacity building, to isolate and identify the PGT from alternate hosts due to the fragility of Aeciospores relative to Uredinospores. So we've just realized that it's not easy to keep the Aeciospores for a longer time. They lose viability. And thanks for listening to me.
Thank you, Ruth. Our next speaker is Dr. Getane Woldayab from the Ethiopian Institute of Agricultural Research in Ambo. So we'll stick with the East African region for now. Thank you. Let me move this.
Thank you very much. Thank you very much. I'm going to talk about what we did in Ethiopia. The title says aecial infections on barberry plants, locally called zinkilla in Ethiopia. In 2000, around 1958, Berberis plant surveys were conducted by the biology department of Addis Ababa University in the northern part of Ethiopia. There were three zones which were surveyed. They were North Shoa, Wello, and Tigray. And later on in 1978, the Plant Protection Research Center conducted Berberis surveys in the North Shoa zone, collected Aeciospores, and infected on wheat seedlings, but they were not successful. No infection was observed on the wheat seedlings. That was in 1978. And in 2009, the pathology section of the PPRC conducted Berberis holstii surveys to investigate whether the plant functions as an alternate host to stem rust in Ethiopia. So these surveys were carried out in the northern part of Ethiopia, Shoa zone. This was one of the Barberry sites, which is found in northern Shoa, northern part of the capital city, Addis Ababa. You can see many Berberis plants, and one of our technicians is collecting Aeciospores from this place. And in the northern Shoa zone, we found 5 locations where Berberis holstii grows, and the locations range from 2,781 to 2,895 meters above sea level. And there were abundant Aeciospores starting from September to January, and after January, the amount gradually declines, and you will not get any Aeciospores after January. February, April, May, you may not get Aeciospores. So these leaves show the Aeciospores were abundant, and you can see the Aeciospores as many as 3, 4, 5 per leaf. And our greenhouse inoculation, we have received seeds from CDL, and we used 10 entries, 5 on wheat, 2 barley, 2 rye, and 1 oat. And these were the ones we used for inoculation. This is how we started our inoculation. These are the big tubs, and the smaller tubs are there. The big tubs, we received them from CDL, and the smaller tubs, we did it ourselves at home, and they were covered with wire net. And you can see the seedlings composing of 10 entries, the wheat, the barley, the rye, and the oat seedlings. The packets are filled with infected leaves of Berberis with Aeciospores. Here, this slide shows how we can place the infected Aeciospores, infected leaves with Aeciospores on the wire net. Here, after doing everything, we place them in the humid chamber. The chamber is made of polyethylene sheet, and we have placed all the materials in the humid chamber, and including the humidifier. Here, after 24 or 48 hours, you can see yellow pustules, yellow Aeciospores on the leaves. This is what we got, and after drying the seedlings, gradually we put them in the greenhouse. So we have tried so far, we have tried 6 inoculations, and we obtained the following. There was no infection on McNear wheat, Morocco, Lemhi, and on Otana oat, there was no infection at all. There was 1 infection on line E on wheat, and a few infection on SR31, and many infections on prolific, the rye and barley. We got many infections. When we do the inoculations, we always have a negative control, and the negative control tells you that whether there is contamination or not. So we have tried to compare the infections on each line. So conclusion, our results suggest that Berberis holstii functions as an alternate host to stem rust of cereals in Ethiopia, but this result is preliminary and will require confirmation and further study. I have an acknowledgement. I wish to acknowledge BGRI DRRW project for financing this activity and giving me the opportunity to participate in this technical workshop. Inoculation materials and research supports were generously provided by CDL. Thank you.
Thank you very much for that. Our next speaker, we're going to move out of the East Africa region into Central Russia. Our next speaker is Dr. Skoletnova from Lomonosov State University in Moscow in the Russian Federation.
Good morning, everyone. First of all, I'd like to say thank you for all persons from organizing committee to my participation in BGRI. Thank you. Now, to address the questions which Iago has posted, I'll try to give a brief description of Berberis status in Central Russia. Okay. The aecial sampling was performed in the narrow region around Moscow and Tver areas. You can see red dots. And one of the first questions was, are there any Berberis in the region? Yes. And some of them are susceptible to stem rust. It's common for Central Russia when the Barberry bushes are planted near the crop fields. What we have, for example, in Golitsyn city, where the All-Russian Research Institute of Phytopathology, I'd like to show the point of institute where we study. Yeah, here, the point. Yeah. There have no outbreaks of stem rust on wheat in Central Russia for the last 40 years, and it's possible the reason is the resistance of wheat cultivars and wheat genotyping, which performed by our colleagues from the Institute of Plant Protection in St. Petersburg will soon answer whether it's correct or not. Returning to Berberis, in Central Russia, they infected in May, and early June. Then, since summer months, we observed infection on barley and wild grasses. Yeah. Occasionally, some wheat cultivars are infected moderately, like we had this season in August. Yeah. Okay. Some words about our sampling strategy. We consider more devices. We consider allocation of bushes as a sample. It may be a single bush or group of bushes which are the same species or next to each other. We suppose that freshly collected Aeciospores are more effective to inoculation than those which have been stored. Using susceptible cultivars, we isolated rye, oat, and wheat stem rust pathogens in the aecial samples from Berberis vulgaris and Berberis purpurea. We indicated that they are susceptible for stem rust. Next. Yeah. For identification of PGT races, we use the international set of wheat stem differential lines. From 2000 to 2009. About 20 different races were isolated. They are maintained at the Moscow State University as a collection of pathotypes. Here you can see the frequencies of PGT races, and there are races with different numbers of virulence genes in the aecial samples. Yeah. So the viral spectrum of viral spectrum on Berberis is rather wide. So this suggests that the sexual cycle may contribute to diversity of stem rust in central region. And summarizing my presentation, Berberis species distributed in central region of Russia were shown to serve as alternate hosts for stem rust pathogen under natural conditions. Diversity of stem rust races was isolated from these species. Yeah. Thank you very much.
Thank you. Now we're going to broaden the discussion a bit beyond stem rust to other Proxinea species. And so that'll take us to Sweden and Dr. Berlin from Uppsala University.
Swedish University of Agricultural Sciences.
Oh no, that's wrong. Which university?
Swedish University of Agricultural Sciences.
Swedish University of Agricultural Sciences.
Yeah.
There you go.
Okay, thank you, Jago, for the introduction. So today I will talk to you about the first part of my PhD studies, and that is about a stem rust population that is clearly in the connection with the alternate host. The project came about when a man who is working with the Sea Service in Sweden noted that the increase of stem rust on oats has increased, and he thought it could have some connection with the increase of barberry bushes since the repeal of the law of barberry education. And since the emergence of the GD 99, my supervisor Jonathan Yuan thought that would be a neat system to study to try to understand what would happen if the wheat stem rust came to Sweden. So, the Swedish law of barberry eradication was repealed in 1994, and on the map you see the incidence of barberries, and this map is from a web page called Artportalen, which is a web page to which hobby botanists can report any plant that they find when they do surveys. So this is only based on people who are interested in plants that have reported. You can find this for any plant, and you see there are some spots where there's not so many dots, like here and this here, and that's just because there is no reports. It doesn't mean that there is no barberry. And the main wheat growing areas of Sweden is here, where you have this really purple area, and it's also here, but here in the south, very close to Denmark, where you find mogens, here is not so many barberry bushes. Yeah. So to introduce you a little bit about how the stem rust cycle look in Sweden, I would have to guide you through the agriculture of the year in Sweden. And of course, you know, it's a little bit in the north, it's a little bit chilly in the beginning of the year, so nothing is growing. And you start to plant the spring sown crops in April. And the first leaf of the barberry will be seen in the beginning of May. And you find the first acia maybe in the end of May, but more usually I find it in the beginning of June, and then it continues during the summer. And the first Aeciospores of rye, you will find the first ones in the end of June, the last week of June. And on oats, you will find it from mid-July. And of course, as the crop mature, the shift goes to Aeciospores. And usually I see that in the second week of August. Of course, it depends on where you are and in the very long country of Sweden, but this is approximately where you see it. And then you have the harvest of both fall and spring sown crops, and then in September you sow the rye and wheat. And this makes that it's a forced population. The population of stem rust has to go through the sexual cycle for the oat to complete to be able to infect. But for rye, there might be some overwintering. And if we look at the data that I got from my collections, this is one study where I collected 30 samples in in some rye and oats field, and at the same time I took samples from barberry bushes, and I analyzed them with 11 SSR markers, and made a lot of analysis. And this is one which is a structure analysis, and this each color shows the common ancestry. So you can see that I don't know if you can. You see the rye is it's very blue, and you can see blue in the barberry populations. And the oats, you have purple and yellow, and you see some purple and yellow here. And you don't see, okay, you have something yellow, purple here, but they are very different, the rye and the oat stem rust. And they also meet on barberry. So it is quite clear that they meet on barberry, but it doesn't seem to mate, so there must be some type of reproduction barrier between the two. And I don't only see this in the molecular data, you can also see the differences between the rye or wheat stem rust and the oat on aecial morphology. And if you want to see that, you can look on my poster. Okay, so when I did my collection, I did it in a grid like this. It was 10 meters apart between each sample. And if I took 30 samples, I got 30 genotypes. And so it's a very, very large diversity within the stem rust population. And the number of genotypes, do they reflect the number of races? I don't know. So, yeah. So to summarize this, we can ask ourselves, why are there no stem rust on wheat? Because we have the barberry. We have severe epidemics on oats, and you can see some on natural grasses as well. We have some stem rust on rye. And there is a large phenotypic variation both within and between fields. And Pathogen Variability is clearly completing its sexual cycle in Sweden. So the two main questions here now is, does the population identified as Pathogen Variability Forma Specialis Triticea ursicalis lack the virulence necessary to infect the wheat? And does wheat grown in Sweden have effective resistant genes? And maybe the two questions are the same coin, one version for the pathologist and one version for the breeders. And that was what I wanted to say.
Thank you very much. Okay. Now we're going to bring the story home to China with Dr. Kang. He is based out of the Northwest SciTech University of Agriculture and Forestry. He's going to speak to us about the relationship between the alternate host and wheat stripe rust in this area.
Would you like this here? Yeah, sure. Okay.
I'm so short.
Yeah.
Good morning, ladies and gentlemen. It's my pleasure to give a short report on the research progress on tenant host and the sexual stage of wheat stripe rust in China. As you may know, the wheat stripe rust is a destructive disease throughout all the winter wheat regions in China, and is considered the most important disease of wheat. Based on the historical epidemiology data for the wheat stripe rust, the wheat growing region in China can be divided into 3 areas. This is over summer area and over winter area. This is east epidemic areas. In the mountains west areas, stripe rust can over summer on the volunteer wheat seedlings and later mature spring wheat. So this is the mountains in the over some areas. Here, what we looked is exactly the volunteer seedlings. You see a lot of Inoculation on the wheat. So each year autumns, the Inoculation spread from west to east every year after year. So the west over summer areas has catched much international and national attention and considered as hotspots for the emergence of the new races of wheat stripe rust in China, because most new races are first detected in this region in recent history. A high genetic diversity within the region's PST population has been reported by different research groups. The genetic recombination was found to occur for wheat stripe rust in this region. However, at this moment, we don't know the reason why the west over summer areas become the hotspots for wheat stripe rust in China. As we may know, the virulence variation for rust may be due to sexual hybridization, mutation, and somatical hybridization. However, the mechanism of the sexual hybridization for wheat stripe rust has been neglected since the sexual stage was prone to be absent. However, in 2010, some Berberis species are shown as attempted host for wheat stripe rust pathogen. This report gave us very limitations because one of the species is a Chinese species. So we ask ourselves, does the sex stage of the wheat stripe rust occur under natural conditions, particularly in China? To answer these questions, maybe we should ask another 3 questions like here, like mentioned by Iago. So to answer these questions, we should do something. First, we make the field survey for Berberis in China. As we know from the literature, there are 500 Barberry species described in the world. In China, we have 215 species. So From this table, you may know most of the Chinese Barberry species are distributed in the west region. Look at here, yeah. And lots of Berberis are distributed in the west over some areas. So this photo we taken from the west over some areas, such as in Gansu, Sichuan province. So you can see different Berberis coexistence with wheat fields. Here, I think everyone knows this big man here this summer in my areas. So, also some Berberis distributed in the mountains nearby the fields. So now you see, it's very common to see Berberis in a field. Now maybe you will ask question, are these Berberis susceptible to wheat strap rust? So we must answer this question. So we collect seeds and the seedlings of Berberis species from the field and inoculated Eustenia of Pixinia sweetforms in greenhouse. Normally, we can see the pycnia on the leaf of susceptible Berberis 11 days to 14 days after inoculations. Here shows some infection of the pathogen on the leaf. About 20 days after inoculation, we see aecial develop on the leaf. So this is aecial. And Aeciospore can infect the wheat leaf through stomata and produce the symptoms of strap rust. So at this moment, we 22 species have been tested. We found 16 species are susceptible to the wheat strap rust, at least here in this table. So among of the 16 species, some species are even green, distributed in the southwest regions, such as Sichuan province, Yunnan province. Others are deciduous, distributed in the northwest region, such as in Gansu province and Shanxi province. Some susceptible species, such as this one and this one, are widely distributed in the west over some areas. So now you will ask, are any Berberis infected by strap rust and the natural condition in China? So it's common to see the aecial on the Barberry in the fields. So last year, we collect 3,600 infected Barberry leaf in the field and inoculated wheat with Aeciospore in the greenhouse. So what happened, we will see. Anyway, most of the sample did not give any symptoms. Fortunately, we get 4 strap rust culture from 3 Berberis, like here, yeah. So our PCR test used 2 PST-specific primer pair demonstrated recovery over 4 culture of wheat strap rust from the Berberis. So this table shows a comparison of infection type of on the Chinese different hostels of 8 major races and PST and 4 PST culture recovered from the 3 Berberis species. So this is 4 cultures isolated from the Barberry. This is 8 major races. So from this table, we can see the infection type of 4 Barberry-derived culture are different as compared to the major Chinese races. Now maybe you will ask another question. Does the sexual stage and natural condition contribute to the variation in virulence for widespread rust in China? To answer these questions, we make 8-2 single-uredium isolates from recovered culture from the aecial. Then we test its the virulence on the difference, like here, yeah. This table shows the virulence difference among single-uredium isolates from single-uredium derived culture. This one, on difference. So from this table, we can see high diversity in virulence among single-uredium isolates. You can see from, so this, for each of them, the virulence is different. So this table shows the same results for another culture. Yeah, this one. The results is the same. So from our results, I give a primary summary for that. So far, 16 of the 22 tested Barberry species from over some areas so susceptible to widespread rust. This indicates that there is a great diversity of potential alternate hosts for widespread rust in China. For culture of widespread rust are obtained from the 3 Berberis species in the field. This means the sexual stage of widespread rust occurs under natural conditions in China. And the virulence test for single-uredium isolates recovered from the aecial demonstrated the sex stage contributes to the variation in virulence for widespread rust pathogen in China. But anyway, so according to our results, now we draw such a life cycle for the widespread rust in China. So it should include a sex stage on the wheat and also a sex stage on the Berberis. So anyway, what we talked today is just primary results from last 2 years. I think for future, we should do much more for in this aspect. I think more Barberry species need to be tested for the susceptible to the widespread rust in China. And I think it's most important for us, more field survey need to be conducted. And more evidence is needed to elucidate the relationship between the genetic diversity and the sex staging of widespread rust in China. So anyway, it's a lot to do for this. Yeah. So finally, I will give to the thanks to my colleagues, Dr. Zhao Jie, Zhang Hongchang, and other students and teachers in my group. And also Dr. Xian Min, Jin Yue, and other friends in international cooperation. And also thanks to the financial support from different projects. Thank you very much.
Thank you very much. Now I'd like to invite Dr. Nazari to come up, who's going to speak about the potential role of alternate hosts in Central and Western Asia. He's from ICARTA. Thank you. Let's move you forward.
Good morning. In this presentation, I will try to present and share with you the preliminary results that we have started last year with a small fund provided by DRRW project to see and get some information about the role of alternate host in yellow rust and the UG99 pathway. The countries that they were involved, my role in this project was coordinations, providing the technical support and also the survey in some of the countries. And also we had a collaboration with Aarhus University in Denmark for genotyping of the samples. Dr. Dave Hudson for using the information, and Dr. Jens in Aarhus University to put information in the GIS system and the toolbox, I hope. And from the countries, we work with Iran in the Seed and Plant Improvement Research Institute, and also Plant Protection Research Institute in Iran to collect the samples and get some information. In Georgia, Dr. Zoya. In Azerbaijan, Ms. Kenul Aslanova and Dr. Mohteshem. And then in Pakistan, Dr. Atiga Rahman. And in Uzbekistan, Dr. Amir Manov and Zafar Zayov, and also in Tajikistan. So what we tried in summary to do, to collect the samples, to do the survey, and also to do some work, DNA work in Aarhus University. And in the end, to have some kind of specifications and also taxonomy to see if the samples that we have in this region, belong to, they have the Poxinia infections, and which species is yellow rust, is a stem rust, or other rust on the grasses. So in general, we have collected samples, all the region that we did the survey in Iran, in Uzbekistan, in Azerbaijan, Tajikistan, and Georgia. Everywhere, first of all, we have the Berberis. We don't know the species. This is one of the main tasks that we need to look after. The distribution of the Berberis bush plants is high, especially in the highlands, in some region up to 2,000 to 500 to 2,400 or 2,700 meter above the sea level. The species, they are different according to the morphology of the plants. And then almost in all places we have seen the Berberis infections. But it was very interesting during the 2012 and also some activity in 2011. Despite the high infection of Berberis, we did not see a stem rust except in few locations that still we are not sure is coming from Berberis or not. But high infection, Berberis infection, we have seen it and we have recorded. But yellow rust was not existed at all, even in case of Azerbaijan, not in the whole country. So they collected their samples. The survey was done in the regions. Then we have collected the sample. You will see the samples from Pakistan, from Iran, and from Georgia. We try to document, the samples. We try to document the samples and record to track the samples from sampling, from GPS, and to the molecular lab. So in one of the places in Seed and Plant Improvement Institute, my colleagues Dr. Patpour and Dr. Afshari, because they have the facility, they have tried to recover the aecium infections from wheat. One of the major issues that we should really take care of it, and we should consider it in the glass house, is to be sure that the infection that we are getting from in the glass house is coming from Berberis, not from greenhouse contaminations, especially in the rooms in the glass houses that you are working with the stem rust, with the leaf rust, and other rust. Growing the plants clean before the inoculation is very important. The methodology, and this is something that we hope that after these discussions, we will come up with a common and a standard protocol like for yellow rust. So what we did, we collected the samples. Sometimes we just take the plants, grow them in the glass house, we grow them. This cage, we designed it to grow the plants under the cage from the beginning, from the sowing, before inoculations. And the inoculation, we have used different methods of inoculations. One of the things that we try to do, washing the plants, the infected Barberries leaf, wash them, and then incubate them for 24 hours, then put them in petri dishes and in humidity chamber, collect the spores by rubbing or putting the oil, spores in the oil using the inoculations, or just putting the plants under the infected Barberries branches. So in the end, we got some infections. We used barley, and unfortunately we didn't have a full set to use the other host, and this is a result for race analysis. Stem rust. One samples we had the yellow rust. Still we are working on it, and we are not sure that samples was from Barberry infection. But these samples, if we assume that we didn't have any contaminations, that we cannot actually at this stage to say 100% the samples that we were using, they were from Barberries. But it's very interesting, the race as a TT-TTF, TT-TTC is very common in Iran. And if you see that they have a virulence for SR36, of course they don't have virulence for SR31 and 24. And so the difference between one of the race, TT-SSK, that is low in combination 7, YR17, and it was low on SRTMP. Usually the Iranian stem rust population, they are high on SRTMP. But in that lab, we have UG99. We have TTK-SK, that is virulence for SR31. And so this is something new in our lab. What we tried that it is virulent for SR31, so it may be its contamination is coming from UG99. Then it is avirulent for SR17. It is not UG99. So this kind of information is just the beginning of the work that we need to do more investigations and to have a better facility to prove this. We have collected the samples from our colleagues in this region. We have sent the samples to Aarhus University, and Dr. Anna Maria Justison, she has tried to DNA extraction and doing the PCR with the markers. And these markers, they show that they have some potential to differentiate between the species. And they have sequenced the samples. And so far, what we have found that we have two main clusters. This is only the preliminary data. We are trying to work with more samples. We have Poxenia graminis, Poxenia astriformis, and within the Poxenia graminis cluster sequence shows that 94 to 95% identity to PGT sequence in the gene bank. Also, all the sequences within the Poxenia astriformis clusters are from Tajikistan. From the other regions, so far we could not see any signal or indications of the yellow rust, Poxenia astriformis triticid. And then these samples from Tajikistan, they show about 99 to almost 100% similarity to the Poxenia astriformis dactylis. So, and then none of them, they were identical to Astriformis triticid. So for the molecular work, the future is we need to do more sequencing, more sampling, and then to use maybe different markers, and to get more reference sequence from the grasses to compare between the different species. And also for the surveillance, we had only very small grant. Maybe I should say we had only $10,000 to do the survey in 5-6 countries. But it doesn't mean that this money did the survey. The problem in the survey that we used the existing BGRI surveillance protocol and the fund provided for the rust. But the problem is that the time for surveillance in Berberis area is different from wheat. So we need to have more fund, more people, skilled and standard protocol for the survey. Also, taxonomy is very important. Taxonomy of the Berberis species in that region is unknown. So we don't know which species. That's why we try to do the documentation, but I think we have some expertise internationally and also to region to help us for this. And with these beautiful plants, I will finish my presentation. Berberis is not only a troublemaker for rust, and it's very beautiful crop, a cash crop in Iran, and I hope that one day you will have the taste of the Berberis when we have the Iranian rice dish. Thank you very much.
Thank you very much. Our last panelist on this rather large panel is Dr. Eugene from the Cereal Disease Lab at the University of Minnesota, and he's going to give us an overview of gaps in knowledge.
Thank you, Iago. Thank you for everybody coming to this session. I feel my colleagues present some very, although preliminary, but very exciting results. I'm really glad. In fact, some of them first time I saw them and I heard them. The chair left me left the good part for me, which is very depressing. Is in terms of knowledge gaps and challenges. From previous presenters, probably with a couple of exceptions, you can sense that we really are in the dark age. And both in the questions we try to ask and the methodologies we are using. But those are essential biological questions, I think, become very apparent why we need to ask them. And I don't feel kind of outlier by asking those questions. Many of us, one time or another, run into statements like this. Cereal rusts are the most researched plant diseases, or large body of literature exists in cereal rust. We are indeed very proud as a rust pathologist, and we are grateful the predecessors accumulate those large body of knowledge that build the very much foundation of plant pathology principles and the foundation for understand disease residence and breeding for disease residence. However, somebody help me? But you could also interpret in the other way. If you work with rust for a few years, you really want to take those statements back. And you could interpret that, in fact, some of those statements telling us there are not too many mysteries left and not very much to do. And indeed, we have lots of things to do. Couple of recent findings from my own research, such as the Strabras life cycle, the left corner, and the Mahonia supporting a local rust population of the right corner. And it's a good demonstration that we really are in a deficit of deep hole in terms of the knowledge of the rust pathogens. This, at least this part of the deficit. The very question we try to ask today, does Barberry play a role in pathogen variation disease epidemiology in stem rust, stripe rust? I think that's very questions tell us we are very poor in knowledge. And I want to use a couple of examples that we have experienced when we look into the kidney questions. First question, in fact, very beginning when we walk into First time I was in Kenya, 2005, after the sudden alarm. And the very question we first asked, are there barberries? Yes, record said there was, but you talk to anybody there, nobody knows what the plants look like, where they are, and where to look. And fortunately, Ruth has a very capable crew, and we will find that thing quickly. And species indeed is fairly common in Kenya. Second question, you will see the recurrent theme of the question we try to ask. It's very similar. Is this particular species susceptible to stem rust? It turns out this is a very easy question to address because you can, through greenhouse inoculation, address this question very quickly. But I dare to ask probably no more than a handful of people in this audience is ever inoculated barberry with rust disease. And that tells you we are really, another aspect really in deficit. Like Ruth stated, we are really stuck in the last question, which is the central question to address the overall question. Does barberry function as an alternate host in Kenya? We don't know yet. And there's many issues encountered by us trying to solve this question. It's the inoculation technology, very immature in my opinion. How long Aeciospore can survive, what specific inoculation conditions we can manipulate to maintain the reliable inoculation, we don't know. But there's a success like highlighted by Dr. Khan and by Dr. Katani. So you can make it work if you try hard. And next question is really when somebody like me work with a group of folks out to the field to find a barberry, really happy, joyful. When the next question comes, what species is? Everybody stare, everybody else try to find the answer. And really, this is a relatively easy only in Eastern Africa because that's the only species report. I challenge this group that to look into this might be wrong, but we assume that's correct. And we know in Eastern Africa you find Barberry holstii. But when colleagues in Pakistan, Nepal, India, Bhutto, and many other places like Nagari, Sydney, Central Asia, when you run into Barberry, and nobody really knows what it is. And I think this piece, the black hole seems getting bigger and deeper. The reason is we don't have the taxonomist in Barberry is extinct species, as many of us know. And very few places you can find taxonomist support. I don't have one. I have to look at the keys myself and look very agonizingly slow, try to kill the species. I just can kill less than half of species. But imagine Dr. Khan has to deal with 200 plus species. So the issue is can we develop a robust assay to identify Barberry species? Well, in the age, you can do thousands of varieties genotyping by sequencing. You can do the same thing by isolates of rust. Why we need to forget Barberry? And I have been presenting this question to a number of colleagues. I have not run into a brave soul to take up this task. I don't know what would be the issue that prevent us to looking to develop a robust diagnosis technique. You can pick up the species quickly. I think we need that knowledge, not just for the pure interest of taxonomic or phylogeny, but for the purpose of helping us identify solving the rust problems. And we really need that. Jeez, it's so slow. This is our last slide. I want to tell you that Anna Berlin told you that Barberry is coming back. As Yago, through the introduction, told you, we have put so much effort, money, and worldwide, particularly in Europe, in North America, put so much effort in eradicating them. When you stop doing that, they have tendency to come back. That's a biological thing, have magic to themselves. And in North America, we experience them coming back quite fast. In Minnesota, Wisconsin, places we surveyed, and Pacific Northwest, where we have trouble with stem rust epidemic still ongoing right now. We have Barberry issues, eastern United States, particularly New England, we have huge Barberry problems over there, and they hybridize naturally all the time. So I think the picture, we are trying to still chasing a few elementary questions before we can get the answer. I'm afraid bigger questions are going to come up very soon that we need to address the Barberry issue coming back of Barberries. I think Anna raised a particular very important issue with resistance, why we didn't get stem rust in Sweden. And I'm wondering what UG99 would do there if that thing's in that population. So in fact, worldwide might be seeing similar situation. Even you have Barberry infection, like I already said, you don't have stem rust in wheat. I would think probably our resident package is still effective even to the Barberry populations out there. Let's see, I would have one more slide. I want to remind my colleagues remain to be skeptical. For the reason that I think that although the results are very exciting, but it's very preliminary in nature and need yet to be confirmed vigorously, and lots of experiments need to be done. And this is for one reason I want to precaution you. So don't get too overexcited with what the progress-wise. Secondly, I have been working under skeptical crowd for a while. I feel that a very strong motivation to drive us to do better science. So with that, I kind of give back to the chair.
Okay, thank you. I'm just going to give just a few words of summary here, and then we'll bring all the panelists up for questions with the time we have left. So as Dr. Hodson said yesterday, UG99 was a catalyst. I liked the language of that. It was a new virulent race that made us realize how complacent we had become to the threat of wheat rust in the latter part of the 20th century. Now, since its formal inception 7 years ago, the GRI, now the BGRI, has made tremendous progress toward its original stated objectives in terms of research, capacity building, the deployment of resistance, really preparing nations for the arrival of the UG99 race group. As these races march onward to ever wider geographical reach, we also have the opportunity to look ahead to the next leap in virulence. How do we prepare for it? As our panelists have outlined for you this morning, there's reason to think that at least one prong in our strategy for achieving increased durability of resistance should entail a greater understanding of the role of the alternate host in wheat-growing regions around the world, particularly in historic hotspots of novel race emergence. Though work on the alternate host has lagged behind somewhat, progress has been made in recent years. The discovery of new species of alternate hosts, the discovery of natural sexual populations of stem rust in Africa and North America, and of stripe rust in Asia, direct evidence that the sexual stage on barberry can lead to increased variation in virulent in both stem and stripe rust. History has taught us not to be complacent with breeding for rust resistance, and it has also taught us that we should not be complacent with regard to the alternate host, a message that gains even greater significance in light of the rebounding populations of common barberry in North America and Europe that Eugen just referred to. So, some progress has been made, but we are in the dark ages still in a lot of ways, and work remains to be done. So that's one of the reasons we've just produced a Barberry surveillance training video. It's now available through the BGRI website, as well as on the CD in your registration packet. It's about 20 minutes long, and it provides some background regarding barberries and demonstrates a method, still a basic method that's in its early stages, but a method for isolating cereal rust species from infected barberry leaves. This training video, incidentally, dovetails with the BGRI's earlier video on race identification, and we hope will expand the work of the rust research community. So with that, I'd like to invite all the panelists up here so you can open the floor for questions. We have a few minutes anyway. Thank you. Okay, so while everyone's getting settled, does anyone have any questions to fire away? Xianmi.
Well, first, I'd like to make a clarification for your comments about the Pacific Northwest of the U.S. about the barberry eradication. So, in the Pacific Northwest, they started this barberry eradication in the middle of 1940s and ended in 1978. So this Washington State was actively participating in that eradication. So thousands of the barberries were removed and treated. But yes, that's one of this Idaho State was just partially involved in that eradication. So even though the barberry, and another factor is there's a barberry, mainly there's a barberry, Berberis, there's a European barberry, or we call common barberry, that was introduced to that area. And nowadays, there's not a barberry area. It's still very hard to find a barberry. So far, we're only able to find less than 30 barberries in Washington and northern Idaho. It'll take quite a bit of effort to find other plants, but these are not very common growing plants in that area. And the second comment about this, now these barberries serve as an alternate host for both stem rust and scrub rust. When we do this inoculation, we always need to keep in mind these two different species. They are require different temperatures for inoculation. So either you can do different temperatures inoculation, or you try to use the temperature that overlap for both species. If you do this inoculation in a very low temperature, you can recover scrub rust mostly, and you can occasionally catch stem rust. But if you do this inoculation in a very high temperature, over 18 degrees and to 23 degrees like reading, you cannot recover scrub rust. Mostly you just recover stem rust.
Thank you very much for those comments. Just to say, the training video does get into some of those technical details with temperatures for isolating the two rusts. But thank you for the surveillance question, or the eradication for Barberry and Northwest. Any questions?
I have a question for you. when you were talking about the gaps in the knowledge on Barberry, you mentioned perhaps the need for more taxonomic support. My question to you is this actually what you want? And the reason why I ask it is with gene sequencing technologies now, when they started to take a relook at eucalypt species in Australia, they found actually the taxonomists got it wrong. Now this upsets them an awful lot, but they had to reassign the number of eucalypt species to another genus entirely. Do you really want that, or would you like some of the tools of biotechnology to be your help rather than a few more taxonomies? It's a bit provocative, but I thought we need to tease that out.
Well, I appreciate that. It's working. I appreciate the question, and from our perspective, particular crowd like this here, we do not care what support we get, as long as it tells us what they are. And I think the question to me is for old-fashioned person like me, take flora description to the field, still very helpful, rather than catch a PCR machine, you know. But however, I believe there's a really serious issue with Barberry in terms of trying to sequencing it. I don't have experience. Some of my colleagues can comment. I think quite a few other groups tried already. Perhaps that's why we're lagging behind. In terms of species identification, I think I applaud the current approach, try to put things in different groups. As we saw earlier, Waph dactylis or beloved strains from species of Strabras put into a new species. This is based on molecular evidence. And we have seen this repeat again in many others. But the group, I think, related to particular Barberry, did some Aeciospores-based phylogeny. Surprising to me is nearly most, if not all, species sit where they're supposed to be. And that maybe sequencing could tell you a different thing. But I think at least ITS tells us the old Aaron did her thesis in terms of classification of Barberry was surprisingly correct.
Yes, please.
We actually tried to, in the study that Anna worked on, we actually did sequence the ITS region of the Barberry plants, and it was not clean. In fact, one of them matched very well to something that was reported in GeneBank as magnolia. And I said, that's very strange, that didn't look like magnolia. And then I realized it was probably something that was mahonia and had been misspelled in GeneBank. But what I really wanted to say was that we shouldn't forget the work that Tor did back with flax rust, where he established that virulence was actually recessive, if you want to use his terms, or that the ADR genes were dominant. And this means that if you have individuals that are virulent on SR31 and they're mating with each other, their progeny are also going to be virulent on SR31. And we should keep this in mind when we're looking into the evolution of these new races. We call them UG99, but they may really be descendants of each other.
Thank you very much. Next question.
Yes, so a question is in my mind about this topic, the Barberries as an alternate host. I was thinking that sometimes now eradication comes to the table, and they have done earlier. But my question goes to the panelists, if you eradicate the Barberry completely, do you think that this rust is so smart can find another host and evolve? You put pressure on this pathogen, so evolve to another, for example, the alcohol. So what do you do in that case?
Yeah, we would like to field that one.
Let me try first. That's an excellent question. We have experience with this eradication and virtually got rid of stem rust. That happened in Western Europe. Western Europe stem rust disappeared. I don't think it's due to resistance. And in fact, if you look at varieties residence, particularly Dr. Robert Park, most recent effort did that, and the residence wasn't complex. Nothing to be proud of, I have to tell European colleagues. Yet, stem rust disappeared from Europe a long time ago. North America, in entirely different type of circumstances, get rid of Barberry from the Great Plains, eastern for Rocky Mountains. And sex population become asexual. And we saw steady decline for races like Yago present, but eventually we came to the point the populations near extinct. When only one race remain, and that race happened to be from Pacific Northwest. So I think lack of sex really bothers the fungus. They won't be able to survive without sexual cycle. Even though in eastern United States, we do have substantial susceptible host populations, wild grasses, barley, and other things susceptible to stem rust. So I think when you get rid of the host, sexual hosts, you can in fact contain the population very well. Those are two large, very clear evident examples.
Does anyone else on the panel want to add to that? Yes.
So that's a very proven example that when you remove the alternate host, of course, you reduce the rate of sexual recombinations. But you have to keep in mind that in all these regions that we have visited, we have Berberis, and we have seen for the long time aecial infections, but we have not seen the stem rust epidemic. That's another question we have to keep in mind. At this stage, the question is that Berberis is really alternate host and has a role in evolution of the races of stem rust and stripe rust or not. First of all, we have to answer this question. Then after that, probably we can look for eradications. I think the most important question is that all of us got excited after the current finding of the Berberis as alternate host of the stripe rust. But does it really has a role in Buccinias reformis? If you look at the epidemics in the last 20 years, the epidemics in agriculture, I mean, when you go science for science, you can find the new races. But those new races, they had the role in agriculture. If you look at the epidemics of yellow rust in the last 20 years, we had only two groups. Virulence for two genes, YR9 and YR27. So there are a lot we need to look at it. But I think at this stage eradications is probably will not be the case, especially for this region, but we need to prove, we have to have data for this. Thank you.
Thank you. So we have run over time, been very patient with the large panel. We have time for just one more question. Yes.
Okay, thank you. Okay, the results from China seems exciting. It's a very good result. That means we need to concentrate in that area. So far we have we got comprehensive results in that area. So I think we need to work together. I mean, so far the standardization of the nurseries and this identification is important to collaborate because it seems that China seems to be really ground for yellow rust, while Uganda for stem rust from what we have seen so far, the diversity of the Berberis and the results. The other thing what we need to consider is to include more species. For example, we need to revise according to the result from Dr. Hidak, for example. His isolates could infect rye and wheat, but they are different from species so far. That means in the future we need also to revise the taxonomy position of the pharma species, position of the pathogen. So probably we need to include more the species, grass species, while we are inoculating from Aeciospores to
it's not only that it gets. That's what I have.
Thank you very much. Anyone like to respond to those comments? Yes.
I guess I can. I think. Should I push here? Okay. Oh, it's up. No, you can hear me. Okay. Yeah, I think if you remember my slide with the many colors, it's quite clear that there are wild grasses also supporting the stem rust population. And I think that's true all over where you find barberry. And of course, there is not only stem rust on barberry, or stem rust and stripe rust on barberry. You find many other species.
Okay, thank you again.